Hello my dear students, in this video we will explore how fish float, that is all about the swim bladder. Basically swim bladder in fishes, in most of the fishes it's a characteristic sac-like structure or bag-like structure, and it is situated or it is located between the alimentary canal and the kidney. This structure is called various names such as swim bladder or gas bladder or air bladder. In our present discussion, the name of the bladder is followed by swim bladder to avoid the confusion. Now let's start. The swim bladder is an internal gas-filled organ found in most bony fishes or osteichthyes fishes. It helps the fish control its buoyancy, allowing it to float or sink without using much energy. The main function of the swim bladder is to act as a hydrostatic organ, helping in maintaining buoyancy. The swim bladder evolved from a small outgrowth of the digestive tract or the alimentary canal, and it played an important role in the evolutionary success of bony fishes because it basically acts as a hydrostatic organ and it is responsible for the floating behavior or the movement behavior of the fish. Now study homology between swim bladder and lungs. The swim bladder is located in the same position in the body as the lungs of higher vertebrates such as mammals and birds. Due to its similar position and function, the swim bladder is considered homologous to the lungs, meaning both organs evolved from a common ancestral structure. Key differences between swim bladder and lungs. What is the origin? Swim bladder develops from the dorsal or upper wall of the gut or alimentary canal, and lungs basically develop from the ventral or lower wall of the pharynx, that is the throat region. Blood supply. Swim bladder supplied by the dorsal aorta and lungs is supplied by the sixth aortic arch. Origin and development of swim bladder in fishes. Scientists have different views regarding how the swim bladder develops in fishes. Development in teleost, modern bony fishes. The swim bladder begins as an unpaired pouch or diverticulum growing out from the dorsal or dorsolateral side of the esophagus. Initially it is a small bud-like structure. This pouch then divides into two parts, left part and right part. The left part usually shrinks and disappears except in some primitive fishes. The right part becomes larger, takes a central or median position and forms the main swim bladder. Development. Development in dipnoans and polypteridae, that is in primitive fishes. In the fishes, the swim bladder is modified into lungs. These lungs develop as outgrowth from the floor of the pharynx. Later, these structures rotate around the right side of the alimentary canal to move into a dorsal position. Because of this rotation, the original right lung appears on the left side, just as the torsional effect found in mollusk. Basic structure of the swim bladder. Swim bladder in fishes varies greatly in structure, size and shape. Fundamentally the swim bladder is a tough or hard bag-like or sac-like structure covered with a network of blood capillary or tiny blood vessels. Just below these blood vessels there is a layer of connective tissue called the tunica externa. Beneath this is the tunica interna, which mainly contains smooth muscle fibers and a special gas secreting tissue called the gas gland. The swim bladder is located below the kidneys, between the reproductive organs and above the digestive tract or gut. It may stay connected to the esophagus throughout life, or this connection may disappear in adulthood. Now we study the gas composition of the swim bladder. Biot in 1807 and Morian in 1876 have shown that the gas secreted by the swim bladder is mostly oxygen. Apart from oxygen, the swim bladder also contains nitrogen and a small amount of carbon dioxide. The composition of gases in the swim bladder varies among different fish species. In salmon and related fish like salmonids, nitrogen makes up the largest portion of the gas in the swim bladder. In many other species, the swim bladder contains a mixture of mostly oxygen and carbon dioxide. Types of swim bladder. Swim bladder in fishes can be classified into three types based on the presence and condition of the ductus pneumaticus, a tube connecting the swim bladder to the esophagus. The first type is the Physostomus type. Ductus pneumaticus, in the diagram you can see ductus pneumaticus is present between the swim bladder and the esophagus. Swim bladder in this type develops from the esophagus and it is found in bony ganoid fishes, dipnoans and soft red teleost. Blood supply from ciliaco-mesenteric artery, here blood is supplied, and venous return to heart via vein that joins the hepatic portal vein. This is the derivation of the swim bladder of the fishes from the gut. A diagram indicates the stages of formation of Physostomus type of swim bladder, and image B shows the stages of formation of the Physoclistus type of swim bladder. Now we'll study the Physoclistus type of swim bladder. Ductus pneumaticus is absent in this type. It can be closed or it can be atrophied. It is found in spiny red teleost. Physoclistus type swim bladder contains gas gland with reti mirabilia that is anteroventrally located and it secretes gas. Oval, it is derived from the ductus pneumaticus. It's a postero-dorsal gas absorbing region. Ciliaco-mesenteric artery also a dorsal artery supplies blood here, and gas gland is innervated by the vagus nerve and sympathetic nerve supplies nerves into the oval region. Now let's study the transitional type which is found in the eel or anguilla fish. Intermediate between transitional type is the intermediate type between the Physostomus and Physoclistus types. Ductus pneumaticus is retained here and it become enlarged and forming a chamber with the oval. Gas glands are present in this type. Blood supply from ciliaco-mesenteric artery via a vessel joining the posterior cardinal vein, that is venous return, and it represents a transitional stage in swim bladder evolution. Mechanism of action of swim bladder in fish. The swim bladder helps fish maintain buoyancy by adjusting gas volume. Gas is secreted into it by the gas gland and absorbed by the oval. In Physostomus fishes, gas is regulated through the ductus pneumaticus, while in Physoclistus fishes, it occurs via blood vessel. Gas secretion, oxygen diffuses from blood into the swim bladder, and it is controlled by capillary network called reti mirabilis. Gas resorption, it occurs through via oval, and oxygen is absorbed from the swim bladder back into the blood. Thin-walled and vascularized region facilitate or help gas diffusion. Function, increase gas with the swim bladder, fish becomes lighter in weight and it rises in the water, and decreases gas in the swim bladder, then the fish becomes heavier and it sinks down. So when swim bladder fills with oxygen or gas, fish becomes lighter and floats up. Swim bladder slightly deflated, then fish remains at the same depth, and when swim bladder is very less oxygen, fish sinks to the bottom. Here the picture shows how the reti mirabilia maintain the oxygen concentration within the swim bladder. Modification in the swim bladder. Swim bladder varies in size, shape and function in different kinds of fishes. It is absent in adult shark, rays, bottom dwellers and deep sea fishes. A temporary form may appear during early development. In flat fishes, it is present when young but shrinks as they settle to the bottom. In shark embryo, a small outgrowth from the foregut may be seen. In some sharks and rays, tiny pits are found in the esophagus near the fifth pouch. Modification in Physostomus swim bladder, polypterus or beachy. Swim bladder has two lobes. Right lobe is longer, opens into pharynx via a slit-like glottis with muscular control. Inner lining is very smooth, partly ciliated, walls are muscular and rich in blood vessels. It is supplied by pulmonary arteries. Blood drains into the hepatic vein. In case of lungfishes or dipnoans, swim bladder functions as a lung, alveoli for breathing. Neoceratodus has a single lobe. Protopterus and Lepidosiren has two lobes. It is similar to lungs of land vertebrates in structure and function. Sturgeons or Accipenser, small oval swim bladder. Ductus pneumaticus opens into the gut behind pharynx. There is no glottis. Duct closes by narrowing, and only the right embryonic lobe develops into the adult bladder. Emia lepistosius এর ক্ষেত্রে কি দেখা যায়? Emia and lepistosius, there is a swim bladder is long unpaired sac along the body cavity. Left lobe appears briefly during the development but it disappears. It connects the esophagus through a dorsal glottis. Walls are vascular and circulated. Blood supply in case of amia, pulmonary arteries. Lepistoceus ক্ষেত্রে dorsal artery supplies the blood. Left ductus cuvieri sent nerves, sent blood in the vein. Lepistoceus ক্ষেত্রে right postcardinal vein. Gymnarchus arteries from the third and fourth gill arch merges to supply both pulmonary and ciliaco-mesenteric arteries. It represents a link between typical swim bladder and lung in case of Gymnarchus. Clupea, Hertzians and others, Pelona or Carnax. Front duct connects to stomach. A second duct at the back opens near the anus. Rare and unique modification is found here. Now we'll study modification in Physioclister swim bladder. Gel structure, it begins as a physiostomus in embryo, becomes Physioclistus in adult. It is composed of two chambers, anterior and posterior. It is connected by ductus communicans. The aperture is regulated by the muscles. Special adaptations: posterior chamber with reta mirabili may flatten and form a oval in families like Myctophidae, Parsidae and Mugilidae. The oval is thin-walled and highly muscular, specialized for gas reabsorption, controlled by circular and longitudinal muscles, important for fishes with rapid vertical movement. Weberian ossicles. Weberian ossicles are a chain of small bones like tripus, intercalarium, scaphium and the claustrum. Here is the diagram. That is a chain of Weberian ossicles separated, found in some teleost fish like carps and catfishes that connect the swim bladder to the inner ear. They transmit vibrations from the swim bladder which act as a resonating chamber to the inner ear, enhancing hearing sensitivity under water. Now we will study the functions of swim bladder. It can be act as a hydrostatic organ. We know that it maintains buoyancy by adjusting gas volume via ductus pneumaticus. It is found in physiostomes, or it can be done by diffusion, which is found in Physioclistus fishes. Swim bladder can act as an adjustable float. It enables fish to stay at any depth with minimal effort by regulating body density. Gas strips within the bladder help balance and control movement, act as center of gravity. Respiration function as lung in some fishes for breathing air in case of dipnoans. Swim bladder can act as a resonator. It amplifies sound vibration and transmit them to inner ear via Weberian ossicles. It is responsible for sound production also. It produces sound by air vibration or muscle contraction. It is found in Polypterus, Lepidosiren, etc. Now it is complete. Thank you for watching this video. All the best, my dear students.